Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to everyone watching us today. A very warm welcome to all participants from Malaysia, the SEAMEO Network and affiliated countries to IMR-SEAMEO Webinar Series 2025. The theme today is Decoding Rare Disease using Bioinformatics, and our presenters today are from the Institute for Medical Research in Malaysia. I am Julaina Abdul Jalil, the head of Inborn Areas of Metabolism, IEM and Genetic Unit, Nutrition, Metabolic and Cardiovascular Research Center, IMR, and I will be the moderator for today's session. To start this session today, I just would like to do a brief introduction about what is IEM and what is our unit, IEM and Genetic Unit, doing in IMR. Just a brief introduction on inborn error of metabolism, IEM. It is a rare genetic or inherited disorders in which the body cannot properly turn food into energy. It is usually caused by defects in specific proteins, usually enzymes, that helps break down metabolized parts of food. And the overall incidence or cumulative incidence of IEM is 1 in 2,500 of birth and affecting all races in the world. And IEM is categorized as rare disease. And IEM usually affects babies and children, but also can occur in adulthood. In our unit, our efforts have been focused in the development of new potential therapies, particularly to IEM and early onset genetic diseases, biochemical and molecular profiling, bioinformatics, machine learning, and AI tools, which will then be discussed by our two presenters later, development of disease models, and approach for new screening or detection methods. And IEM and genetics units missions are to develop high-quality research for IEM and genetic disorders for the betterment and cost-effectiveness of patient care. To become a training center for technology transfer and provide consultation services for IEM and genetic disorder. This slide is to show what are the research areas we are focusing on, which are glycobiology, neurotransmitter disorders, mitochondrial disorders, lysosomal storage disorders, and also other genetic diseases, particularly the early onset ones. And this next slide is just to show what are our activities in our units. The session of taking patient sample, the activities in the lab, and also what we do in collaboration, and also our participation in a lot of other activities. So just to say that we play hard, but we work harder. And I think that ends the introduction to IEM unit. We want to invite for further collaborations with all the participants that can collaborate with us today. Okay, ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on biomedical research, and present research outcomes. Before we begin the presentations, allow me to announce a few housekeeping messages from the Secretariat. First, number one, we will not be taking questions during the presentations, but audience may post your questions using the link shared in the YouTube channel, and we will be taking those questions after both presentations have ended. An attendance link is provided in the YouTube channel for those who would like to receive an e-certificate of attendance. And don't forget to subscribe to our National Institute of Health Malaysia YouTube channel and click for notifications whenever there's a new post. So ladies and gentlemen, for today's webinar, we have two speakers. Our first speaker would be Dr. Muhammad Khairul Nizam Muhammad Khalid, and I will do a brief introduction about him. He is a research officer in IEM and Genetic Unit. He recently graduated with PhD from University of Tasmania, Australia. Since his return to Malaysia, he has worked very closely with clinical geneticists to help revisit patients' exome data, particularly those with negative or inconclusive findings. He is also interested in applying artificial intelligence and advanced computational tools to solve variants of unknown significance, VUS, and perform periodic exome reanalysis. Nizam believes that genomics has a great potential to transform lives, and he hopes to showcase this through his work. For today, Dr. Muhammad Khairul Nizam will be sharing his research experience with the presentation titled From Exomes to Molecular Diagnosis, Identifying Genetic Variants. Dr. Nizam, the screen is yours.
Thank you, Dr. Julaina, for the kind introduction. Bismillahirrahmanirrahim. Assalamualaikum and good morning, everyone. My name is Muhammad Khairul Nizam. The title of my talk today is From Exomes to Molecular Diagnosis, Identifying Genetic Variants. So let's begin with a brief overview of rare diseases in Malaysia. Patients with rare disease often manifest severe and debilitating conditions, as we can see here in the media coverage, showing patients with retinoblastoma on the left, Prader-Willi syndrome in the middle, and spinal muscular atrophy on the right. And these are among more than 6,000 different rare diseases identified to date. 83% of all rare genetic diseases affect children. Even though we have more than 6,000 different diseases and they are individually rare, but collectively they are common, affecting more than 300 million people worldwide. And a staggering 80% of rare diseases are caused by abnormality in the genome, which means they have genetic origin. Recently, we achieved a great milestone for the country, where the first national policy for rare diseases was launched. This policy covers many aspects, such as early detection and screening, access to treatment and supportive care, research and innovation, capacity building, and the call to establish the National Rare Disease Registry. On the ground, we have strong collaboration between lab scientists and clinical geneticists. And over the years, this has led to development of tests and methods to help diagnose patients with rare disease. And as a result of that, we have published key papers in the field, and I've shown just a few of them here today. So, why is molecular diagnosis important? Well, it is important because it provides the answer to the patients in their families. So through molecular diagnosis, we'll know which gene is responsible, which genetic variant is pathogenic, and this will help to end the diagnostic journey of a patient. And through this information, we can help to improve the disease management and facilitate familial testing. So now, other members of the family can be tested for the same genetic variant. And this will also help in genetic counseling. In some cases, diagnosis help to open the door to more effective treatment. And now, with the advent of gene therapy and precision genome editing, some of these patients may be eligible for clinical trials involving these orphan drugs. So before we move further, let's briefly talk about genetic variants in general. So the key message here is that pathogenic genetic variants disrupt the genetic code. So here is some simple example to illustrate this. So we can have a substitution where one base, such as T here, is replaced to A. This is single nucleotide change, or SNV. In other cases, we can have insertion where C and T nucleotides are being added, or one nucleotide such as T is being deleted. Or we can have some complex changes such as insertion and deletion happening at the same time. We call it indel. So how is that affecting our genome? So let's imagine our genetic code is like a sentence. The sky is blue. So when we have a single nucleotide change, so the sky is blue is now changed to the say is blue, which doesn't make any sense. But this is the nature of genetic variant. It disrupts the genetic code, leading to production of faulty protein, and this finally results in genetic disease. And this gets a lot more complicated when we consider pathogenic variants can come in various different sizes, ranging from short SNV and indel that we saw before, less than 50 bases, to copy number variants, such as shown down here, where a stretch of DNA can be duplicated or even deleted altogether. And this range all the way to more severe and catastrophic structural variants, where large segments of the chromosomes can be either inverted or translocated to another part of the chromosome, causing massive changes to the genome. There are also other variant types that we need to consider. So we know in our cell, we have the nucleus and the mitochondria. Nucleus will have its own nuclear genome, whereas mitochondria will have its own mitochondrial genome. So some variants are located on this mitochondrial genome, and these mitochondrial variants are responsible for mitochondrial diseases. And there are also this unique type of variant, which we call short tandem repeats, where, for example, in this gene, we have a normal person having CGG of less than 55 repeats, and this gene is expressed in a normal way. But in a patient, this CGG number or repeat expanded greatly to more than 200. And this led to instability to this part of the genome, leading the body to effectively shut down the expression of this gene, and this lead to the disease. And this type of variant is called STR, or short tandem repeat variant. So let's put all of this together. SNV, CNV, STR, mitochondria. They contribute to different degree to the problem. But most of the variants responsible for disease is of SNV or indel in nature, about 80% of them, followed by CNV and SV, STR, and mitochondria. So it is important for us to recognize this and consider this when we are doing testing. So the bulk of our SNV and indel was traditionally detected by using a method called Sanger sequencing, which now we're going to go a bit more detail. So Sanger sequencing is based on Sanger chemistry, which uses fluorescent chain terminating dideoxynucleotides for sequencing reaction. So we have these fluorescent dyes labeled at the end of DNA, and from this fluorescent dye, we can convert this into the DNA sequence. So one key limitation of this technology is that every piece of DNA that you want to sequence needs to be separated in separate tubes, and therefore it makes the sequencing very low throughput. So let's have a solid example of this through, again, our example with retinoblastoma. Retinoblastoma is a very aggressive childhood cancer, which is caused by mutation in this gene called RB1. So for your information, this study was published about 10 years ago by our group, and today we're going to go a bit deeper into the results. So RB1 gene is a very massive gene. As you can see here, this is the structure of the gene. It's a very large, and all these vertical lines indicate the exons, and all these long stretches represent the introns. So in genetics of human, we are interested in the exonic region because they code for protein. So when we see the RB1 gene was broken into 27 exons, this means that we need to have at least 27 different reactions to cover the entire gene. We did that, and we identified pathogenic variants across different parts of the gene. So some in exon 18, some in exon 21, some in exon 4. In fact, we proceed to CNV analysis using MLPA for this cohort, just to make sure we cover all the possible genetic variants in this gene. So even though we were able to identify the genetic variants, we realized that this method does not scale well, especially if we have a lot of patients. Imagine for one patient times 27 exons, we need to do 27 reactions, and this may take like a month to complete. How about if you have 50 patients times 27? That's more than 1,000 reactions to perform, and it will take many, many months to do. And this explains why when you send your sample to a genetic lab, the result may come back in a few months rather than a few weeks. So as the demand for genetic testing grows, we need a method that is scalable and high throughput. So a take-home message for the first part of my presentation is that rare disease disproportionately affect children, and investigating the patient's DNA would reveal the genetic abnormalities underlying the disease. And this is why we focus on rare disease so much. This pathogenic variant can come in different forms and sizes, from small SNVs to large CNV and SV to unique mitochondria and STR. SNV and indel is a major contributor to rare disease, and Sanger sequencing was a gold standard, but it suffers from being low throughput and slow. A combination of different techniques are necessary to get a good yield, So in our case, we combine SNVs and CNV using two different methods to achieve this. So next, to overcome this bottleneck that we described, we move to a next generation of sequencing technology called whole exome sequencing. So whole exome sequencing is based on this next generation sequencing platform, or NGS. I believe many of us have heard about NGS in the past. So NGS takes a radically different approach to sequencing, where instead of starting with one piece of DNA, one whole genome was broken down into smaller pieces. And all of these fragments are sequenced together in a massively parallel reaction. So this high level of multiplicity achieve high throughput and scale that we need to perform this level of analysis. So NGS, we have two main popular applications. One is whole exome sequencing, the second is whole genome sequencing. So as the name suggests, whole genome sequencing is sequencing the entirety of the genome content. So we cover the entire exon, the introns, and everything in between. But for whole exome sequencing, we're only covering the exonic part of the gene. As we described earlier, exons are responsible to code for protein, and therefore it makes more sense to cover this part of the gene that we understand better, because it's easier to interpret, easier to analyze, and it's cheaper. And this is the method of choice for our lab. So now let's briefly talk about the workflow for whole exome sequencing analysis. So we start off with a patient sample. So either we start with a blood sample, or now we can even access swab sample for sampling. And from these biosamples, we extract the total genomic DNA and we prepare them for a sequencing reaction. So once the library is ready, we put it into the machine, and this machine will generate the millions of DNA sequences that we expect from NGS. And all of this data is being put together into a raw data file called FASTQ. So next time when your friend talks about FASTQ, you know what it is. It is a raw sequencing data from NGS. So what's inside the FASTQ? FASTQ is a very big data, often ranges in gigabases in size. So please do not open this with your standard notepad. So what I'm showing here is a screenshot of a FASTQ running on the terminal. So here we can see our familiar DNA bases, A, T, C, and G, and this line, each line represents one DNA molecule that was sequenced. And this line extends for millions and millions of rows just to show you the amount of data that is generated in one single reaction. So with whole exome sequencing, we are generating this high throughput data, and the challenge now is to analyze this in a systematic fashion. And this is when we turn to bioinformatics. So from raw data, now we move to doing alignment of the data. And this alignment is important because we know this data was originally scrambled. It has no order to them. So to make sense of the data, we need to map the data back to where it came from, which is from the human genome. So this process is called mapping, and we have a selection of human genome to choose from, either HG19, HG38, or the most recent telomere to telomere, TOT to T in short. So through this mapping process, we will convert our file from FASTQ to a file called BAM. So when we hear about BAM, it means that it's an aligned data. And we can perform this mapping step through various different ways depending on the resources available to the lab. So if your lab doesn't have bioinformatician and you have the resources, you have some funding to support the work, then you can choose to subscribe to Dragon, for example. Because Dragon offers a simple interface where you can drag and drop your sequencing data, your FASTQ, put it on the cloud, and just with a few clicks, your analysis is on the way. But if you have resources, you have the computer infrastructure, you have bioinformatician around, then you can explore something like GATK and SAREC. So let's now go a bit further into using command lines. GATK is Genome Analysis Toolkit, which is the most widely used bioinformatics pipeline for whole exome sequencing. Even until today, if you read the literature on whole exome sequencing, you'll find GATK somewhere in the methods. It is such widely used, such that development of this pipeline has been aggressively pursued. And recently, a group of bioinformaticians formed an NF core community. And the aim of this community is to standardize and simplify execution of complex bioinformatics pipeline. So they developed a pipeline called SAREC, which is used for whole exome sequencing, and the link is provided here. So what it does is remove the headache from having to install various tools, managing different versions, and having issues with incompatibilities. But in order to run GATK and SAREC, we need to have access to Linux operating system. But lucky for us, now Windows have the option to install Linux environment as an application on your system through installing what we call as WSL, or Windows Subsystem for Linux. So WSL allows a full Linux environment to run natively on Windows without very sophisticated setup. But you need to have a certain specification for your computer, such as you need to have certain version of Windows to run it. But if you do have and meet the requirement, then installing just as simple as running this code here, WSL -- install, and if everything goes well, you end up with a screen like this. And then you're ready to run your first code. So let's move back to SAREC. So if you look into the SAREC web page, you see, like I said, it simplifies the execution. So instead of running lines and lines of code, SAREC has made everything so simple such that the basic script looks like this. So with just one line of code, you can convert your FASTQ to BAM file. But this can be extensively configured based on your need. If you have more sophisticated analysis, you can of course add more to this code to make it more suited for your analysis. And if everything goes well, your screen will show up like this, and your data is currently being processed for you. Now we live in the age of AI. So AI agents like ChatGPT are widely used for processing text information. But from my experience, I found that Claude AI provide the best solution when it comes to coding. So here's an example of me giving a prompt to Claude AI saying, give me a script to run NF-core SAREC on a single exome. And in a matter of seconds, Claude AI produce a full pipeline that I can then copy and use to process my data. But of course, all this requires some knowledge of command lines. And if you want to get started, I would recommend to go to the sandbox.io and then explore the interactive tutorials that are available there for you to learn the basics of Linux before you embark on this. Now let's go back to our data. So once we have the aligned data from the mapping step, we can actually explore this data further using a software called IGV, or Integrative Genomics Viewer. So when you open the software, you put in your data, you will get something like this. So remember, the vertical lines are your exons, and all your data are clustered, are squeezed around these vertical boxes. Again, confirming that this is an exome data, not some other data type like whole genome sequencing. And what you can do is you can start exploring your data. You can move from exon to exon, from gene to gene, just to look at what you have. And as you explore, you can see these lines start to appear in your data, from your sequencing grid at the bottom all the way to the top. And this indicates the location of potential variants. We call this raw variants. So this indicates the position where your patient data, which is up here, is different compared to the reference data down here. And this is the genetic variant that we are looking for. So when we zoom further into these lines of different colors, either you can get a line of one color or a line of two colors. So the line with one color indicates is a homozygous single nucleotide variant, in which both of the patient allele has been changed to another base. In this case, A in the reference has been completely changed to G, giving an A to G homozygous variant. But in another case, we see two colors, which means there's two different nucleotides here. So we still have the original reference here as one of the allele, and the second allele has been changed to G. And now you have an A to G heterozygous variant. So we can see these raw variants in our aligned data. But to use it, we need to actually do much more filtering to get only the high quality ones out of this. And this step is called variant calling. So let's go move to variant data. So again, now we have BAM data, and we want to do variant calling to get our variant data, and this variant data is stored in a file called VCF. So next time you heard about VCF, you know this VCF will contain all of your genetic variant. And we can do this again with the same 4 or even the same tools that we described earlier. So let's open the VCF up in any notepad because the size is quite small, and we can see these lines and lines of information. Each line represents one variant. So in a typical exome, we expect about 20 to 30,000 variants in one sample. So if you go down the list, you get about 20 to 30,000 lines in this file. So these are the variants that have passed stringent filtering step to remove any technical artifacts and low quality variants. So let's put this together now. So we have BAM down here, and we can actually load the file, the VCF, in the IGV to have a composite view of both. So you can see here, for example, in BRCA1 gene, you have 1, 2, 3, 4, and 5 lines in the BAM indicating potential variants, and all of these 5 lines now appear in your VCF, which means these 5 variants pass the filter and now appear in your VCF. But there's a line down here that was evident in your BAM, but it was not found in your VCF. And this means through the variant calling step, this variant was dropped because it was located outside of your exonic region. So this is how variant calling extract the variant out from your alignment. So now let's put everything back together. So from raw data to mapping, from FASTQ to BAM, and from aligned data to variant, which means from your BAM to your VCF. But in fact, you can actually combine the mapping and variant calling step, so you can get from your FASTQ to VCF in one go. So your VCF will contain SNV and indel, which we discussed before is the most common variant type for RD, for rare disease. So how do we get then from our variant data to the final answer? So out of these 20,000 to 30,000 variants, only a few is actually responsible for disease. For example, for autosomal dominant disease such as retinoblastoma that we talk about in this presentation, only one pathogenic variant in RB1 gene is needed to achieve a diagnosis. For autosomal recessive, we need 2 pathogenic variants in one gene to get a diagnosis. And the remaining thousands of variants that we saw are part of normal genetic variation in human, and these are benign. So how then do we find this a needle in a haystack? So one way to do that is to combine the genotype information that we get from our variant data or the VCF with what we saw in the patient or the clinical symptoms. This phenotype is described in a systematic way using human phenotype ontology, because we need to convert things like fever, dysmorphism into a language that the computer can recognize, and we do that through the use of HPO. So when we combine the genotype and phenotype, it will enable us to prioritize the variant. So which means the genotype that best explain the phenotype will be listed at the top of the list, and all those unrelated benign variants will then be moved out to the bottom of the list, allowing us to focus on the relevant ones. And one popular command line tool for this purpose is Exomizer. So once we perform this variant prioritization step, we'll discuss with clinical geneticists to see if the variants that are listed on the top of the list are actually relevant to the disease, and before we decide on the final answer. So that's it for SMV and indels. But how about the rest of the variant types that we discussed earlier? Mitochondria, STRs, CNVs. Well, yes, we can actually detect those variant types also from one exome data. But we need to use specialized bioinformatic tools for this purpose. For example, for CNVs, there's a tool called CNV Kit. For mitochondrial variants, mToolbox. Repeat expansion using Expansion Hunter. Structured variants using Manta. And you can run MELT to get a new type of variant, which I also just learned about, which is mobile element insertion. And these tools are all based on command lines. So you can see having expertise in command line really extend what you can do with your data. So let's look at how CNV looks like in an exome. So we have here 3 patients' exomes shown here in IGV. So we have, again, we call this gene A, and we have all of these exons running around it, and we have sequencing read sitting on all of the exons. And in patient 1, you see in these 2 exons, there's no sequencing read on top of these 2 exons. And this actually indicates that in this patient, the patient has a homozygous deletion of 2 exons, which is a class of CNV, which can be seen and detected through whole exome sequencing. So let's go to a case study. So this family appeared in the press about a few years ago, where 4 out of 8 children were affected by neurological disease in their 20s and 30s. Their father suffered the same disease and has passed away. Our clinical geneticist from Penang Hospital, Dr. Cheng Gek Siu, helped us to see this family and organize for whole exome sequencing. So for this work, we use command line tool to identify pathogenic short tandem repeat. Because remember, in STR, expansion of the repeat is the one that is caused the pathogenic effect. So to run this tool, we need a BAM file. Remember, it's an aligned file that we talked about before. The BAM file is the input for Expansion Hunter. So down here is showing the basic script to run Expansion Hunter. It's not that complicated. So if you know command line, this is actually quite simple. So to perform the analysis, you run Expansion Hunter, and what the tool does is that it checks 30 different genes that are known to have STRs in them. So instead of looking one by one, the tool will go through all 30 genes simultaneously and provide a list of the repeat number in each of the gene. And then we can scroll down, compare with the reference, and see which one of them has expansion. So we did that because the initial sequencing of this family, or one of the patients, showed negative findings. And from the presentation, we are suspecting that this family suffers from a disease called spinal cerebral ataxia, or SCAR. SCAR is caused by genes, a few genes, ATXN1, 2, 3, 4, and 5. And then to do this in the lab requires step-by-step testing. So what we did was we put this data through Expansion Hunter, and when we go through the result, we saw a possible heterozygous CAG trinucleotide expansion in one of the SCAR gene, which is ATXN3. So a normal range is between 12 and 44 repeats, and in this patient, the first allele has 11 repeats, which is close to the normal range. But in the second allele, it has 53 repeats, which is far in the pathogenic range for this gene. So once we discovered this, we discussed with the lab, our molecular diagnostic labs, and the team helped us to do confirmatory testing. So what they did was they performed triple repeat prime PCR, or TP-PCR, to check on the repeat sizes for ATXN3 gene in all of the 4 affected siblings. And this is the result from the analysis, showing that the repeat sizes for all 4 patients extend well into the pathogenic range, here as shown here. And through TP-PCR, the repeat size was estimated to be around 70 versus our 53 from Expansion Hunter. But we know this is a limitation of bioinformatics because we use short read for analysis, and therefore it will not be super accurate. But at least it gives us an indication on where to focus on for confirmatory testing. So to sum up, whole exome sequencing can be that one test to cover all different variant types that are important in rare disease. As opposed to before, single sequencing can cover SNV and indels and mitochondria, TP-PCR for STR, MAPA for CNVs, and so on. Now we have one test that can do almost all of this in one go. And this results also in significant improvement in scalability. Because let's compare again with the traditional method where one sample takes about 1 month for RB1 gene, and if the patient is negative, we proceed with MLPA, which takes another 2 weeks. And altogether, 6 weeks will be taken up just for one sample. Versus now with exome sequencing, library preparation and sequencing takes about 1 week, and bioinformatics take a maximum of 1 week as well. But it can be faster because we are dealing with a known single gene. So you can see how much time that we can save from this, and we can then do many more samples with that. But there are also limitations to whole exome sequencing. The most obvious one is that it covers only the exons, and therefore any pathogenic variants in the introns or in the promoter or regulatory region will be missed. So even though whole exome can detect CNV, it has limited power to identify the boundary. So where the CNV starts and ends exactly in the genome is unknown because it has very sparse coverage. Then other variant types such as STR will require validation because we know, as the example shown before, it's not very accurate. So before reporting, we need to confirm it first. But at least it gives you an indication on where to look at. So overall summary, whole exome sequencing is a high throughput and scalable technology that allow comprehensive detection of genetic variants. Bioinformatics allow deeper dive into the exome data, enabling us to extract more information that will be useful to diagnose our patients. And it is important to keep the patient data, especially those with no diagnosis, so that they can benefit from latest advances in bioinformatic tools and also genetic knowledge. So remember, behind every dataset, there's a family waiting for answers. And sometimes the diagnosis is already there. So keep looking. So with that, I would like to end my presentation. Thank you so much for your attention. And back to you, Dr. Julaina.
Thank you very much, Dr. Nizam. That is indeed a very interesting talk where you can see evolution from the Sanger sequencing to the next gene sequencing, particularly WES or whole exome sequencing, which has been presented by Dr. Nizam in detection of the disease causing by genetic changes. So I think I'm sure that all the participants and audience have a lot of questions, but you have to be very patient because we will only answer those questions at the end of the second speaker session. And kindly submit your question using the link provided in the YouTube channel. Okay, so we move on to the next presenter. Next, I would like to introduce our second speaker for today, Dr. Ernie Zuraida binti Ali. She is currently a research officer attached to the IEM and Genetic Unit. She obtained her Bachelor of Science Honours in Biology and Master of Science in Molecular Biology from the University of Putra, Malaysia. She started her career in IMR in 2006 and attached to the Molecular Diagnostic Unit IMR, where then she pursued her PhD study in Pharmacoinformatics at the Faculty of Pharmacy UiTM and graduated in 2020. Dr. Ernie has wide exposure and experience in wet lab and dry lab, and currently her main research focuses are on computational study, which covers mutational effect, drug design, and initial vaccine development. She is also leading the work on developing the newborn screening kit for spinal muscular atrophy, SMA, and she is actively collaborating with academicians and postgraduate supervision with the universities. Through her expertise in computational skills, she also assists clinicians on developing protein structure modeling for mutational study. It is our great privilege to have here with us Dr. Ernie Zuraida to share her research. And her presentation title is Leveraging Protein Structure and AI to Interpret Genetic Variants. Over to you, Dr. Ernie. The screen is now yours.
As we know, every person's DNA is a little bit different. These small differences we call genetic variants. Sometimes the genetic variant changes can affect the functions of the protein in our body. Therefore, by analyzing protein structure and employing artificial intelligence predictions, we can figure out whether these changes might cause problems or not. Bismillahirrahmanirrahim. Thank you, Dr. Julaina, for the kind introductions. Assalamualaikum and good morning, everyone. It's an honor to be here to present on the topics of leveraging protein structure and AI predictions to interpret genetic variants. I hope by the end of my presentations, whoever watch and listen to these sessions will get some idea on how to analyze any genetic variants that you may have by using the tools that I will share later. This is the outline of my presentations. As mentioned by Dr. Nizam, through the Sanger and NGS platforms, we will obtain many genetic variants per patient. The question is, how do we analyze and interpret and classify these variants? Variance interpretations generally follow the SCMG guidelines. However, there is no single standardized workflow. It really depends on the information that available for each variant. Basically, in practice, variance interpretations typically consider the following factors. First, how common the variance in the general populations. If a variant commonly found in healthy individuals, usually we classify it as a benign. Second, how well that part of the gene is conserved across different species. Third, we examine what has been reported in literature or in disease variant databases. We also recommend to do segregation study to provide additional evidence. And if you are lucky, we might have functional study that give us more direct evidence. Out of these approaches, I choose to focus on the last one, which is on computational predictions. They actually have a two approach, which is the traditionals and advanced in silico tools. And our main goal is to classify genetic variants into one of these categories. Negative, which means it's not associated with disease. Pathogenics, that means it's likely to cause a disease. And the last one is variants of uncertain significance, or known as VUS, which basically means we just do not have enough information to say either it's a negative or pathogenics. In silico prediction tools actually have evolved significantly from traditional machine learning to sophisticated AI and molecular dynamic simulations. Traditional in silico tools, such as those using classical machine learnings like random forest, really rely mainly on DNA or protein sequence data and also conservation information. This approach, they provide a fast results. However, they are actually limited to moderate accuracy due to their dependence on the small data sets. In contrast, modern AI tools actually they use deep learning and large-scale structure and variance datasets to achieve higher accuracy and also the scalability. This tool actually can predict the functional impact of variants with greater precision and efficiency, so making them ideal for large-scale and high-throughput analysis. For molecular dynamics or MD simulations like GROMACS and AMBER, they actually represent the most detailed level of analysis. They provide us atomic level view by modeling protein under realistic physical conditions and also offering very high accuracy and deep structural insights. This platform is computationally intensive and is actually best suited for validating key variants rather than for screenings. So, when do we use advanced in silico tools? We use them when we discover a new or novel variants in disease genes, and also we do not have enough experimental evidence to rely on to tell us what it does. Or we were unable to do segregation study, maybe because we do not have access to family samples. In those situations, predictions are very useful. Although predictions is not a final answer, but actually it gives a strong clue and can guide us the next steps in variant interpretations. Now that we know when to use these tools and how they are generally works, let's talk a bit more about what is AI prediction tools. These tools use artificial intelligence, which including modern machine learning and deep learning methods to analyze complex biological data. In simple terms, they are actually trained to recognize patterns in huge amounts of genetic or proteins information that might be too complicated for humans to spot on their own. Therefore, by doing this, AI tools actually can help scientists to predict how certain genetic changes might affect a protein's functions, how changes can affect the structure's conformations, and even the findings from the predictions also can help and assist us to relate the findings with the disease. They actually have several types of AI prediction tools which researchers commonly use. Some of the well-known examples, which includes IF, which use evolutionary data, it actually looks at how proteins have changed or stayed the same across different species to predict whether variants might be pathogenic. ESM4, which focuses more on protein structure, it predicts how a protein folds in its 3D shapes, which help us to understand how a variant might affect the structure. And the last one is AlphaMiSense, which this tool is actually built based on the DeepMind AlphaFold technology. It uses deep learning to predict whether a specific genetic changes is likely to cause disease. Actually, several studies have compared AI prediction tools like IF and AlphaMiSense with various traditional in silico tools such as SIF, Polyfence, and Mutation Tester. The result actually has been quite encouraging, where overall the AI tools show higher accuracy in predicting whether a genetic variant is likely to be harmful or harmless as compared to traditional tools. These improvements actually comes from the fact that AI model can learn from a huge and diverse data set, which help them make more reliable predictions, especially for variants that traditional tools find difficult to interpret. So, how do we actually use these AI prediction tools? The good news is, it's actually very simple and very straightforward. The user just simply enter the gene names or UniProt ID or related identifier to automatically generate the results. Once the information is entered, the tool will process automatically and give you results that to show whether the variance is likely to be benign or disease-causing. The results come in the forms of a score, which actually help us to interpret the potential impact of the variance. In general, the higher score usually means the variance is more likely to be pathogenic, which a lower score that suggests it's likely to be benign. As we know that each of these tools actually they have their own cut-off value. For example, in if tools, variance that have value more than 0.64 will classified as a pathogenic, while variance that has a value less than 0.36 is classified as benign. Here is example of the project that we run on OTC variance. The main goal is to predict the pathogenic impacts of this variance by using the AI tools. And for the analysis, we documented the score produced by the tools, which to achieve the consensus for variant classifications later. Why we use 3 different AI tools? We use 3 different AI tools because each tool actually examines a variance from a slightly different perspective to generate a consensus predictions, which actually is made easier and more reliable to classify them. Okay, so by combining the results, we actually reduce the chance of error and get a more confident predictions. Based on the consensus, okay, we can see that all 3 variants we predict as a pathogenic, meaning they are likely to be a harmful impacts on the protein and could contribute to the disease. So what are the advantages of using AI prediction tools? First, they are actually faster, where user can get results directly through a website without a complex setups. Second, they are scalable, meaning they can handle large data sets with many variants at once. Therefore, they can improve predictions of accuracy. And perhaps the most importantly, they can provide predictions for all possible missense variants, including those that are novel or not yet found in patients, which traditional tools often cannot access. Now, let's move to another advanced in silico approach, which is we call molecular dynamics or MD simulations. It's actually a computer-based method that helps us to study how atoms and molecules move and interact over time. In a simple terms, it allows us to see what happened at the atomic levels. So, how does it work? Okay, in MD simulations, we build a virtual model of molecules in the computer. Then the computer will calculate how each atom moves and interacts with other using the real law of physics. Mainly, they use Newton's law of motions. This means that our simulations is not just a guessing, okay? It's using a real physical principles to predict how atoms behave based on the force like attractions, repulsions, and chemical bonding. So, the result that we get are actually scientifically grounded. You can imagine like the MD simulations like you watch a slow motions of ball in the box. Okay, after that, after we run the simulations, it produces something that we call a trajectory. Okay, it's actually a record every atom positions over the times. By studying this, okay, we can understand how molecules change the shapes, interacts, and respond to different conditions. MD are also incredibly useful, where actually it can assist many researchers in different areas of research that would be impossible to get through the experimental alone. So what skills are needed before we using MD simulations? To run MD effectively, we actually need a good foundations in the few key scientific areas such as computational science, physics, structural biology, and also chemistry. It is important to have at least basic Linux skills since most MD software runs on the Linux systems. In short, working with MD is not just about running a programs. Okay, it's actually about combining knowledge from science and computations to understand how molecules move and behave in a real life. However, don't worry too much about computational skills. To me, as long as you understand the basic concept of MD and once you get the idea behind it, okay, and you have a clear research questions and clear research gaps that you want to do, you will find that it's not complicated as it sounds. Okay, once you have a dedicated and you patience, and of course, I think you can do it. Since MD simulations actually are run using computer, they are actually require specific hardware to work efficiently. This is because MD simulations, they involve a million of tiny calculations, as actually they track the movement of atoms over times. They need a high processing power, large memory, and fast storage. Usually, normal desktops is not powerful enough to do this kind of the works. Okay, that's why we need to use high performance computer or HPC, which is a very powerful system to design to handle a large and complex simulations. Here is actually the example for the HPC that our institution purchased in 2019, which we still use until today to run our MD simulations projects. At that year, we purchased these specifications is considered as a high specs with a 2-time processor, high NVIDIA GPU cards, RAMs, and also storage. There is also another platform that you can use by using the cloud computing. The price and the specs you have to request with the vendors. Okay. So the time required to complete one MD simulations actually does not depend only on the computer hardware. Okay, but it also depends on the size of the protein studied. Based on the table, for example, we can see that a small protein like OTC, which has about 354 amino acids, it takes about 15 hours to complete the simulations. Okay, but a larger protein such as PDHA1 with about 692 amino acids, it can take up about 40 days to finish. So what it means here? It means that the bigger the molecule, the longer the simulations times. So that's why I always I would like to highlight that having a good infrastructure is very important. This is to ensure that our analysis can be done efficiently. To prepare MD simulations, I have summarized into the 5 key steps. Okay, the first one is system preparations. This is where we set up the molecules, and the second is solvations and ionizations. Here we add water and ion to mimic realistic environments. Third is energy minimizations, where actually we aim to remove clashes and stabilize the systems. Fourth is equilibrations, where we actually adjust the temperature and pressure. This is to make sure the system behaves naturally. And finally, the productions runs where atomic movements are tracked over the times. This is to generate the data for the further analysis. Okay, this is what an MD simulation looks like. Okay, we start with a static protein structure. It's like a snapshot of the molecules. Then we simulate by following the key steps that I have discussed before. Okay, through this process, the protein actually is no longer static. Okay, we can see how it moves, how it changes the shapes and interacts with the other atoms over the times. Post-MD simulation analysis that we carry out, okay, we do after the simulation is complete. Okay, at this stage, we actually examine the trajectory data that generate during the simulations to understand how the protein behave over the times. Okay, the good of the MD simulations is actually they provide a qualitative and quantitative results. Okay, we analyze the data based on the basics and advanced analysis. Okay, this is based on the research question and research gap that you want to look. Okay, example of analysis that we can run is the thermodynamics and physical properties. Okay, structure stabilities and flexibilities. We can look into the interactions and binding energy, and also for the energy changes. And also we also can visualize the protein structure. Okay, in the movie form that we can generate also. Okay, here is actually examples projects on OTC variants using MD simulations. The main goal actually was to examine the effects of OTC variants by using the MD simulations. And for the analysis, we usually compare the wild types protein with its variants, okay, to see how they affect the stability, flexibility, and the overall of OTC structure. This is actually help us to predict whether a particular variant might disrupt protein functions and cause a disease. Okay, this is the results. Shows several important differences between the variants and the wild types proteins. Okay, we measure the RMSD, SASA, RMSF. This is, as you can see from the figure, that have results that shows that the qualitative and qualitative results. Okay, and from the figure we can see that the variants have a high values, which indicate that the variance is more flexible and unstable as compared to the wild types. We also proceed with the hydrogen bond analysis to show that the variants have fewer interactions which we can predict that it affect the shapes of the proteins. We also extend our analysis to observe changes in the binding pockets. This is to see either the variants could affect the important regions, especially the active pockets. Okay, from the figure, we can see that the variants cause an important regions of the protein to shift away from its original positions. Okay, so they potentially have to affect the functions of the OTC protein. So this table actually summarized the finding from our MD analysis. Okay, by looking at the result, we can see that all the variants cause increased structural fluctuations and greater exposure to the solvents. Okay, what this means? It means that the proteins becomes more flexible and less stable. They also reduce the number of hydrogen bonds and create a looser binding pockets. Okay, this change suggests that variants are likely to destabilize the OTC protein and reduce its cofactor binding efficiency. Okay, from the analysis, we get a consensus and we predict that all the variants is likely pathogenic. However, a bit reminder for clinical use, okay, it is best to confirm the predictions with experimental data. Okay, please not just rely on the computational predictions alone. Okay, beyond analyzed genetic variants, MD simulations are also used in many other research areas of research, okay, such as drug designs, enzyme catalysis and mechanisms, antibody-antigen interactions, protein-protein interactions, and protein folding and misfoldings. Actually, MD help researchers to understand in depth how the drug interact with the protein. It actually helps most of the research out there to answer their research questions, okay, how the protein folding, how the enzyme works, which these things is impossible to capture through the experimental alone. MD also, they have several key advantage, okay, they let us actually to see how a protein structure changes when a variance is present and show the movement of molecules over the time at atomic levels. It actually helps us to predict a variance, how variance might affect the protein functions. MD is cost effective, okay, because of it can reduce the needs for extensive labs experiments. And actually it helps us and guide us to choose which variants or targets to be focused in focus in the experiments. As we know that we have a list of variants, okay, it's impossible. We need to run all the variants through the experiments. So by using the MD, we can screen first and only the one that we need to proceed with the experiment. MD also can complement to the AI predictions where they give a more complete pictures of the protein structure effects. Okay, and the last one is especially very useful to use for study the rare or new variants where usually the labs or population data may not exist. By combining AI and MD simulation actually brings several important advantages when predicting the variance effects. Okay, it makes predictions more reliable by combining the strengths of both approach. Okay, for AI, for example, it's very quick to get the results. Okay, while MD they show how the protein actually moves and behave over the times. They actually give us a detailed mechanistic view that static protein structure usually cannot provide. Okay, they also help us to connect structure changes to potential functional impact, so we can have a better understand which variants might impact the protein functions. It's also allow us to prioritize which variants to study in more depth, improve efficiency, and also saving the resources. And last one, importantly, this combined approach actually improve the interpretations of variants, bridge computational data with biological insights, and has a potential to guide clinical decisions in the future. As a conclusion, AI prediction tools assist in processing large amount of data and rapidly predicting variant pathogenicity by analyzing sequence, structure, and evolutions. Okay, on other hand, MD also provides a critical mechanistic insights into the functional consequences of variants at the molecular levels and also reveal a detailed effects of each variant. The integrations of AI prediction tools with MD simulation data, they constitute a robust and sophisticated strategy for understanding and interpret genetic variants, especially when the experimental data is not available. By looking beyond the DNA sequence and into the three-dimensional shapes and movement of protein, we are actually help us to gain deeper insights into how changes at the molecular level can lead to the disease. And lastly, this tool have a great potential to become part of standard variance interpretations frameworks. However, as a reminder, large-scale validation is still required to confirm their clinical use. This is the summary of the complete workflow. Okay, as we receive the sample, we process the sample, we run the NGS sequencings, and NGS will generate the data. And from here, we will have a list of variants. Okay, don't worry if you unable to run functional study in the labs, you still have options to the downstream analysis by using computational approach, which, for example, AI prediction tools and also the MD simulations. Okay, before I end my presentations, I would like to take this opportunity to express my sincere gratitude to Director General of Health Minister of Malaysia, Director of IMR, Head of Center and Head of Units for granting the permissions to present this IMR-SEAMEO webinar. Thank you to clinicians for the collaboration and assistance in providing data for research purposes, and also to IMR staff, especially IMR-SEAMEO webinar. Their continuous support and assist have invaluable in making these presentations possible. Thank you for your times and attentions. Hope you find these information very useful and informative. Thank you again, and back to you, Dr. Julaina.
Thank you very much, Dr. Ernie, for the very comprehensive and very insightful presentation. Well, from her presentation, we can see that AI tools with the MD simulation can predict how even a small change to the genetic can cause alteration, and you can predict that it can cause alteration to either protein structure or protein function. So even though bear in mind that those are in the dry lab, right? So, but then you can actually minimize the extensive lab work later when you want to do the wet lab. And I think that is very interesting, Dr. Ernie. Okay, we will now be moving to the question and answer session. Okay, please do not hesitate to send us questions using the link provided in the YouTube channel, and I will try my best to connect to you, to our distinguished speaker through the posted questions. Nevertheless, if the time does not allow us to address all of the questions, our speakers will try their best to answer the questions through email. It seems that we have a few questions from our audience. Can we go through that? Okay. The first question is from Shirley IMR. Hi, Shirley. Okay, the question is, I would like to know, for the first part of your talk on SNV, have you encountered both normal and SNV sequences, and does the percentage of the SNV versus normal plays a role in disease presentation? To you, Dr. Nizam.
Thank you for the question, Shirley. It's a very interesting one. Because like I mentioned earlier, for SNVs, so when you talk about having normal and the variant together, in a true, what we call as heterozygous case, we would expect the percentage to be 50 and 50, so 50% variant frequency. But there are cases where the fraction of the variant is actually less than 50%. For example, we've seen the mutant allele to be as low as 20% or even less. That would indicate a much more unique genetic event, such as genetic mosaicism, which may happen if we don't consider the genetics of disease into account in our analysis. So yes, thank you for the questions. That help us to highlight the fact that variation in genetic, in the frequency of the variant can reflect whether the variant is actually a true germline variant, or they appear to a process which is creating a mosaic variant in the patient. And that is also, in some cases, contribute to the severity of disease. So patients who have a true heterozygous presentation will be more severe compared to the one that have some level of somatic mosaicism in them. So yes, I would say we have to consider the variant frequency, and then we'll match it up with the presentation in the patient to understand what truly happened in the patient. Thank you.
Okay, thank you very much, Dr. Nizam, for the answer. Shall you have any further question? You can personally meet Dr. Nizam in the IM genetic unit. Okay, I think the second question goes to Dr. Ernie. Can this AI and MD simulation approach be applied to any protein of interest? I think this is an interesting question as well.
Okay, thank you, Dr. Julaina, for the questions. Okay, for AI, yes, because AI tools is actually trained based on the large data sets or known sequence. That means the effects of many genetic substitutions have already been analyzed. Okay, even for variant that does not exist in patients, the AI tool also can able to generate the data. Okay, for MD, it can be applied in the case if the protein structure is available in the uniprods, or the structure can be predicted by using any available tools. However, MD actually is not suitable for unavailable protein structure. That's my answer, Dr. Julaina. Thank you.
Okay, thank you very much, Dr. Ernie. I think we move on to the next question for Dr. Nizam. I think this is an interesting question as well. How to decide on using using whole exome sequencing or whole genome sequencing? To you, Dr. Nizam.
Thank you for the question. So this is interesting one. So when you decide to go with whole exome or whole genome, there are a few things that you need to consider. So to me, the most important, of course, is the cost. So if you have access to some funds, I would say whole genome sequencing is better, because in all the analysis that I showed before, whole genome sequencing will generate a much more complete data compared to whole exome sequencing for obvious reasons. But if you don't have the funds and you have a strong suspicion of which gene is responsible, for example, you know, in my presentation I showed RB1. So RB1, the mutations that are responsible are of SNV and CNV in nature, which can be picked up by whole exome sequencing quite comfortably. So I guess it depends on factors like that in making your decision. Thank you.
So I guess you have to know your disease first, and you decide on that, right, Dr. Nizam? All right. I think in a matter of time, we now proceed to our last question to Dr. Ernie. This question is from Soleha of University Putra Malaysia UPM. How do you classify the MD outputs as pathogenic or benign, Dr. Ernie?
Okay, thank you, Dr. Julaina. Okay, that's a great questions. Okay, we have looked at how the protein behave in the simulations. Okay, because MD actually they provide a qualitative and quantitative results. Okay, so we must observe both data. Okay, if a variance causing significant changes like destabilizing the proteins, altering its shapes, or disrupting important interactions, usually we classify or we predict them as a likely pathogenics. For variants that remain stable or behave like a normal versions or like a wild types, usually we classify or we predict them as a likely benign. That's the answer, Dr. Julaina. Thank you.
Okay, thank you very much, Dr. Ernie. I'm afraid that is all the time we have for the questions and answer session. However, any additional questions will be answered by speakers through email. We hope that this webinar has shed light on some research work that we are conducting in IMR and would spark interest and co-initiate research collaboration with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. Again, don't forget to subscribe to our NIH YouTube channel, National Institute of Health Malaysia. And before I conclude, I would like to announce that we will have the next session of IMR-SEAMEO Webinar Series on the 28th of November 2025. So please mark your calendars. The theme for the next webinar is Systematic Review of Dengue Molecular Epidemiology, Protocol Design and Early Findings, Initial Molecular Epidemiology of Dengue Systematic Review. Well, this is a very interesting topic. We hope you can join us at the next session. Until then, many thanks from all of us at IMR. Take care and stay safe. Thank you.